So today mainly we will be discussing about these important points of identification. Now these important points are of low magnification as well as high magnification. ठीक है, what is the meaning? इसका मीनिंग क्या है? तो ऑलरेडी यू हैव सीन दैट व्हेनेवर वी सी द स्लाइड्स फर्स्ट वी सी अंडर लो मैग्निफिकेशन एंड समटाइम इट इज रिक्वायर्ड वी सी अंडर हाई मैग्निफिकेशन. ठीक है, तो इफ यू पुट द स्लाइड ऑफ किडनी अंडर लो मैग्निफिकेशन देन व्हाट कैन बी सीन? Okay, so that is mentioned over here. So I will request everyone to note down these points that will help you to identify the parts of the kidney. So microscopic part, okay, under microscope. Everyone note it down. So first of all, you can see the kidney is covered by renal capsule. So this renal capsule can also be seen under low magnification. Next, deep to the capsule, we are getting cortex, and deep to the cortex, medulla of the kidney is situated. Now you have to understand what is present in the cortex and what is present in the medulla. Now if you see the cortex, mainly we are getting a circular structure. These are called as renal corpuscles, surrounding which there are tubules cut in various shapes. That means especially in case of cortex we are getting renal corpuscles, and along with that various tubules are there which are cut in various shapes. That means you can see TS, you can see LS of the various tubes. Okay, now what are the which type of tubes you can expect to see in the renal cortex? These are PCT and DCT. ठीक है, तो PCT क्या है? Proximal convoluted tubule. DCT means distal convoluted tubule. Those two types of tubes are present. Where they are present? They are present in the cortex of the kidney. तो किडनी में दो चीज़ें दिखाई देता है. पहला क्या है? फर्स्ट इज द रेनल कॉर्पसल्स. रेनल कॉर्पसल्स में क्या रहता है? ग्लोमेरुलस एंड वोमेन्स कैप्सूल. These two things can be seen. Along with that, the tubes can be seen. What are these two tubes? These are PCT and DCT. That means proximal and distal convoluted tubules. ठीक है. Next, how you will differentiate between the PCT, DCT? First point. Second point, how you will differentiate between the cortex and the medulla? This is another point. Okay, because under microscope you have to differentiate, because clear cut demarcation नहीं है कि इतना से यहाँ तक cortex है, इसके बाद medulla है. Like this demarcations are not there. Okay, तो अगर मान लो ऐसा demarcation नहीं रहता है, then how we used to differentiate? Yes, based on the staining properties. Okay, some will take a light stain, some will take deep stain. Based on that we used to differentiate. ठीक है. Now if you see the PCT and DCT, the main difference is in case of PCT, what is the appearance? They appear dark pink stained tubules. Okay, so PCT darkly stained, and if you see the lumen, it is small and indistinct. And the cells which are lining the PCT, these are simple cuboidal epithelium, that also with brush border. Next, in case of DCT, if you see the distal convoluted tubule, it is light stained and the lumen is distinct. That means lumen is regular, proper shape is there. Indistinct means irregular होता है. Okay, so in case of PCT, first of all it is dark stained. DCT, it is light stained. Here the lumen is small and indistinct. That means irregular lumen. Here the lumen is having distinct shape. Mainly it is oval in nature. Okay, so these are some important parameters by which you can differentiate. Next, here what is the lining epithelium? Simple cuboidal epithelium with brush border. Here also same, but brush border is absent. Okay, so these are some points which can be identified under microscope. Okay, like this we can differentiate the PCT and DCT. तो PCT DCT कहाँ पे रहता है? Kidney का which part of the kidney? They are present in the cortex. No, cortex of the kidney. Now we are talking about histology. ठीक है. Next, you will be able to see the renal corpuscles. तो renal corpuscles में क्या रहता है? As I said, tuft of capillaries are there. That is called as glomerulus. And surrounding the glomerulus, the glomerular capsule which is present, also called as Bowman's capsule. In between these two structures, there will be a narrow space that is called as urinary space. That is also present and it can be seen both under low power also and under high power also. ठीक है. So these are some important points which you have to identify, okay, in low magnification, low power. So already I mentioned that the DCT, they are lined by simple cuboidal epithelium. Okay. Next, the number of PCT are more than the number of DCT. Can you tell me why number of PCT are more as compared to DCT? Because if you see the length of the PCT, it is more as compared to the length of the DCT. So in a cut section, more number of PCT will appear as compared to the DCT. And जो PCT होता है, it is in the form of coil. जो PCT होता है वो coil shaped रहता है, DCT coil shape में नहीं रहता है, ठीक है? Next, if you come to the, so that is about the cortex. Next, you come to the medulla, ठीक है? In the medulla, what you can see, light staining elongated parallel lining running tubules, okay, lightly stained parallel running tubules can be seen in the medulla. Now these tubules are made up of what? These are made up of collecting ducts and loop of Henle. Okay, so mainly in the medulla you will not get the renal corpuscles. Instead of that, what you will be getting? You will be getting lightly stained elongated parallel running tubules, and those parallel running tubules are made up of collecting ducts and loop of Henle. Now there are some areas where the ducts are parallel to each other, okay, which can be seen easily, and there are some areas where cut section of those ducts are also seen. Okay, parallel means complete parallel. Cut section means round structures can be seen. Okay, both can be seen in medulla. Now some of them extend into the cortex. Now the collecting duct are lined by simple cuboidal epithelium. Okay, so as I said, two types of ducts are seen, collecting duct and loop of Henle. So how we will differentiate in between them? Again, with the help of lining epithelium, it can be differentiated. ठीक है? तो collecting tubule का और collecting duct का वो क्या है? Lining epithelium, that is simple cuboidal epithelium. And in case of loop of Henle, two types of epithelium can be seen. Okay, depending on which segment it is, thin segment or thick segment. Now the loop of Henle is lined by simple squamous epithelium in case of thin segment. Okay, and in case of thick segment, again it is simple cuboidal epithelium. Okay, next another important point is there. If you see the collecting duct, okay, it is lightly stained, और जो loop of Henle होता है, it is darkly stained. So like this also we can identify the difference between the two ducts which are mainly present in the medulla. Okay, so if you see from practical point of view, these are the points which you have to know, especially in case of kidney. Next, you can see the diagram. So which part is the cortex? See, this part is the cortex. This is under low magnification. This is under high magnification. ठीक है? तो ये वाला जो part है, this is cortex and this is medulla. Okay? So ये high magnification में है, ये भी high magnification में. This is under low magnification. ठीक है? Now, if you concentrate over here, what you can see first, क्या दिखाई देता है? हाँ? Yes, very good. First you have to see the renal capsule. ये देखो ऊपर renal capsule दिखाई दे रहा है, ये देखो, this is the renal capsule. यहाँ पे भी renal capsule दिखाई दे रहा है, ठीक है? So first you have to see the renal capsule. Why? Just below the renal capsule, the area which is present, it is called as renal cortex. It is called as renal cortex. तो ये देखो, ये capsule के नीचे ये जो पूरा area है, this is renal cortex. ठीक है? तो renal cortex में क्या देखना है? What you have to see in the renal cortex? No, PCT, DCT है renal corpuscles. First you have to concentrate on the darkly stained renal corpuscles. Can you see the renal corpuscles over here? See, these are the renal corpuscles. These are the renal corpuscles. ठीक है? तो renal corpuscles are made up of what? As I said, it is made up of tuft of capillaries which are called as glomerulus, and it is covered by glomerular capsule which is also called as Bowman's capsule. ठीक है? तो जो ये tuft of capillaries रहता है, around that Bowman's capsule रहेगा. ठीक है? So in between the tuft of capillaries and Bowman's capsule, a space is there. It is called as urinary space. Can you see the urinary space? Yeah, that can be seen in high power. Low power में भी दिखाई देता है. I will not say that it cannot be seen. See, ये देखो. ये छोटा सा स्पेस दिखाई दे रहा है, राइट? ये देखो ये वाला स्पेस. बट इन केस ऑफ हाई मैग्निफिकेशन यू कैन सी इट क्लियरली. ये देखो, दिस इज द यूरिनरी स्पेस. ओके, सो ये पहले देखना है. नेक्स्ट व्हाट यू हैव टू आइडेंटिफाई पीसीटी एंड डीसीटी. तो व्हाट इज द डिफरेंस बिटवीन पीसीटी एंड डीसीटी? हाउ विल आइडेंटिफाई? पीसीटी इज डार्कली स्टेन्ड, डीसीटी इज लाइटली स्टेन्ड. Next point, in case of PCT the lumen is indistinct, means irregular lumen रहता है, ठीक है. And in case of DCT lumen is regular, oval shaped. Can you identify? तो in case of PCT DCT it is very difficult to identify in low magnification. It is there but देखो दिखाई नहीं दे रहा है ठीक से. For that you need high magnification. Now in the high magnification can you see the PCT? ये देखो, this is one PCT, this is one PCT. Okay, see the lumen of the PCT. ठीक है. Now see the DCT. Okay, so like this you have to identify which is PCT, which is DCT. Okay, see this is PCT, this is PCT. Okay, this is DCT. देखो, this is DCT. So staining is also important, but instead of that it will be better if you focus on the shape of the lumen. For that you need cross section. If it is parallel, means if the tubules are parallel to each other, or if it is in LS, then it will be difficult. Best way to identify is see the cut section, TS. In TS you can understand the shape of the lumen. ठीक है. Now for example, if a pipe is there, ठीक है. Now suppose एक पाइप है और एक दो पाइप है. Now if you take the cut section and if you see inside what is there inside, then only you can see the shape of the lumen. If they are parallel to each other, ठीक है, दोनों अगर parallel है, then what will be the shape of the lumen you cannot guess. और LS में shape of the lumen पता नहीं चलता है. It is difficult. That's why best way is the focus on the TS to understand the shape of the lumen. वहाँ से identify होगा कि which one is PCT, which one is DCT. And second point is staining. If it is PCT, it will be darkly stained. If it is DCT, it will be lightly stained. Next, another important point. Okay. Wait. What is the difference between the lining epithelium in case of PCT and DCT? Both are same. In case of PCT, brush border is present. In case of DCT, brush border absent. Okay, for brush border it is very difficult to see in high magnification also. Okay, but it is present. तो ये हो गया cortex. What is this? ये क्या है? This is medulla. तो medulla में first of all you have to note that you will not be able to see any renal corpuscle. Can you see any renal corpuscle? No. देखो, रेनल कॉर्पसल मतलब दिस आर द रेनल कॉर्पसल्स. ठीक है, ये देखो, दिस आर द रेनल कॉर्पसल्स. सो दिस कैन ओनली बी सीन इन द कॉर्टेक्स. मेडुला में दिखाई नहीं देता है, यू विल नॉट सी. नाउ डू यू रिमेंबर डिफरेंट टाइप्स ऑफ नेफ्रॉन्स व्हिच वी हैव डिस्कस्ड लास्ट टाइम? yes, cortical nephron and juxtaglomerular nephron. Okay, already we have discussed location, function, structure, all those things we have discussed. It is present in this PPT only. Okay, so now I am not going into this. Next, please come to the medulla. Medulla में क्या दिखाई देगा? Parallel running lightly stained tubules are there. Okay, mainly two types of tubes can be seen. What are they? Collecting duct and loop of Henle. How you will differentiate between collecting duct and loop of Henle? Yes, loop of Henle darkly stained होता है, collecting duct जो होता है lightly stained होता है. First, second point, lining epithelium. In case of loop of Henle, दो segment होता है, thin segment and thick segment. In case of thin segment, lining epithelium is simple squamous epithelium. Thick segment में क्या होता है? Simple cuboidal epithelium. Okay, next, in case of this tube, what is the lining epithelium? It is collecting duct. What is the lining epithelium? It is simple cuboidal epithelium. Okay, now please see the so here also some PCT DCT can be seen. Now see, this is the thin segment. Okay, see, this is the thin segment. You see, this is the thick segment. So see the difference between the thin and thick segment. Lining epithelium is different. Okay, next, here you can see the collecting duct. see the collecting duct, and see the thick ascending limb. ये देखो, can you differentiate the staining? ये देखो, ये वाला और ये वाला. This is the collecting duct, this is the thick segment. Can you differentiate? Okay. So this is how you have to differentiate the structures present in the medulla. So in practical point of view, if we study the kidney, it is not that difficult. Only few points are there. If you remember, easily you can identify the slide in exam, and you can write the identifying features also. Okay. प्रैक्टिकल में अगर मान लो किडनी का स्लाइड आया तो इट विल बी फोकस्ड मेनली ऑन द कॉर्टेक्स. जनरली वी डोंट कीप इट इन मेडुला because student used to get confused. What I have seen. So if it is focused in the cortex, first you have to see renal capsule. Renal capsule के नीचे क्या देखना है? Renal corpuscles, PCT and DCT. ठीक है? Only the points you have to write. That also not in sentence because you are getting only 1 minute. 1 minute में आइडेंटिफाई करना है कौन सा टिश्यू लिखना है और 2 आइडेंटिफाइंग फीचर्स लिखना है. ओके. तो what I have felt that by looking at the slide of the kidney under low magnification also, it is easy to identify. But if it comes in the theory, suppose 5 marks ka ki write about the microscopic structure of kidney, then it is very difficult. Okay, next under high power, so low power, high power both I have shown you. Next, if you see the high power renal cortex, large renal corpuscles can be identified, okay, which we have seen just now. The renal corpuscles consist of tuft of capillaries that form a rounded glomerulus, and the outer wall, the glomerular capsule is present, that is called as Bowman's capsule. Next, the urinary space. So I have shown you the urinary space. Okay, between the glomerulus and the capsule can be seen. The urinary space can also be seen. Now mainly in order to see the urinary space we need high magnification. Next, the PCT and DCT, this seen the same way we have to identify, okay, under high power high magnification. Okay, now this is the renal cortex under high magnification. Okay, so this is the schematic diagram, this is the original diagram, original image. Okay, so in schematic diagram you can see what you have to identify. So this is the renal corpuscle. In renal corpuscle you can see what are these are the tuft of capillaries, okay, and this is the Bowman's capsule. So last time I hope you remember I have mentioned that in case of Bowman's capsule 2 layers are present, right? Outer layer and inner. This is parietal layer and visceral layer, okay. So here these 2 layers can be seen. ये देखो. Next, juxtaglomerular apparatus can also be seen, but this in schematic diagram it is easy to identify, okay? But in case of original image, you see it's not that easy to identify the juxtaglomerular apparatus. Next, PCT, DCT can also be seen, okay? So just see the appearance of the PCT and appearance of the DCT, okay, which is lightly stained, which is darkly stained. Now in renal medulla high magnification, what you can expect? First of all, renal medulla shows number of collecting ducts. Either they will be present longitudinally or transversely. Okay, lining epithelium can also be seen easily, which is cuboidal epithelium. Okay, so the boundaries will be distinct in case of DCT. Now the sections of the thin segment of the loop of Henle can also be seen. Now these thin segments, they are lined by flattened epithelium. Okay, so flattened epithelium and cuboidal epithelium can also be identified easily. Okay, if you see the high power image of renal medulla, especially of the thin segment and the thick segment. Okay, so in thin segment is simple squamous epithelium, and in case of thick segment it is simple cuboidal epithelium. So identification can be done easily. Now this is the renal medulla high power. Okay, so this is the schematic diagram. Here you can see blood vessels are also seen. Along with that you can see this is the collecting duct. Okay, collecting duct are also seen, and along with that you can see the loop of Henle. Okay, see this is the loop of Henle, this is the loop of Henle. Okay, and these are the collecting duct. Now if you concentrate over here, ठीक है, देखो ये collecting duct है, ये collecting duct है, ठीक है. और लूपोफिनली, ये वाला लूपोफिनली है. नाउ सी द स्टेनिंग डिफरेंस. ओके, इन केस ऑफ लूपोफिनली यू कैन सी इट इज डार्कली स्टेन. यहाँ पे जो कलेक्टिंग ट्यूब्यूल दिया है इट इज लाइटली स्टेन. Okay, so like this it can be differentiated. So that is all about the kidney. I hope you have noted down the points. When we'll go to the histo lab, at that time I'll ask you those points. Based on that you have to identify. So in urinary system, what are the various parts after kidney? Ureter. Okay, so you can't forget that in urinary system only kidney is there. No, ureter is also there. इसके बाद क्या आएगा? After ureter, bladder आएगा. Bladder के बाद क्या आएगा? Urethra आएगा. That means you have to know the histology of all those parts also. ठीक है? Now in epithelium chapter, in epithelial tissue chapter, you have studied about the transitional epithelium. Do you remember transitional epithelium? Example क्या है transitional epithelium का? हाँ, so that time you have seen the slide of ureter. Now you will study the ureter. ठीक है? So it's good that you till now you remember the lining epithelium. Now in case of ureter, 3 layers are there. First is the mucosa, smooth muscle coat and adventitia. Okay. Now in case of ureter, serosa is not there. Adventitia is present. Why? Because we know that ureter is situated in the posterior abdominal wall. It is retroperitoneal. ठीक है? तो here we will not get serosa. Entirely it is made up of adventitia. Lining epithelium क्या होगा? Very simple, transitional epithelium. Okay. Along with that we are also getting the supporting lamina propria. Next, smooth muscle coat. तो mainly 2 layers are there, and in the lower 2/3 of the ureter, additional layer is added. ठीक है? That means if you see the smooth muscle coat, inner longitudinal, middle circular, mainly these 2 layers are present. But if you go to the lower 2/3 of the ureter, you are getting outer longitudinal layer. Okay. Can anyone tell me what is the arrangement of the muscle fibers in case of GIT? No. Smooth muscle layer. How it is arranged? Inner circular, outer longitudinal. ठीक है? इन केस ऑफ यूरिनरी सिस्टम उल्टा होता है. ठीक है? यूरिनरी सिस्टम और जो GIT, ठीक, दोनों में जो मसल कोट होता है, अरेंजमेंट दे आर ऑपोजिट टू ईच अदर. उतना याद रखो, बाकी हो जाएगा. तो इन मसल कोट, फर्स्ट ऑफ ऑल इनर लॉन्गिट्यूडनल एंड आउटर लॉन्गिट्यूडनल ओनली प्रेजेंट एट द लोअर 2/3 एंड मिडिल पार्ट इज द मिडिल सर्कुलर वन. Another important point which you have to notice in case of ureter is, if you see the mucosa of the ureter, it is folded, and because of the folding of the mucosa, the appearance is star-shaped. The appearance of the lumen is star-shaped. Okay, so mucous membrane consists of, first of all, lining epithelium will be there. 4 to 5 layers of cells are present. Okay, that is transitional epithelium. Lamina propria is made up of dense irregular connective tissue. Now see, mucosa shows numerous longitudinal folds, and due to the presence of those folds, what is the appearance of the lumen? It is star-shaped. So that you have to identify. Next, smooth muscle coat. Now there are some interesting points regarding smooth muscle coat of the ureter. That means if you trace the smooth muscle coat above and if you trace the smooth muscle coat below, then you will be getting some important and interesting points. What are these? First of all, smooth muscle are arranged in 3 layers, that is inner longitudinal, outer also longitudinal. जो middle वाला होता है that is circular. Okay. Now it is very difficult to identify those layers. Means distinct arrangements are not there. जैसे मान लो ये longitudinal है, ये circular है, ये longitudinal है. Clear cut demarcations are not there. Okay. But still these 3 layers are present. Next, if you trace above, the circular muscle surrounds the renal papillae. Okay, if you remember the cortex and medulla of the kidney, which we have discussed last time, at that time I have told you about the renal papilla. तो जो smooth muscle layer है, okay, the circular layer, it surrounds the renal papilla. Now what is the function? Why it is surrounding the renal papilla? It helps in squeezing the urine out of the ducts into the minor calyces. in renal papilla, mostly we see the opening of the ducts of Bellini. So in that particular part, what is present? Circular muscles are present. So what they will do, they will squeeze it. When they are squeezing it, the urine will come out from those ducts, and then they will go into the minor calyx. Okay, so that is also one function, and for that you have to trace the circular coat in an upward direction. Next, if you trace below, the outer two layers are continuous with the detrusor muscle of the bladder. You have studied about the urinary bladder, right? Urinary bladder is very important. That day also I have mentioned. Those who have not studied, please study about the urinary bladder. Okay? Now in urinary bladder, you know that the smooth muscle of the urinary bladder is called as detrusor muscle. ठीक है, तो जो outer two layers होता है of the ureter, if you trace below, it will continue with the detrusor muscle of the urinary bladder. Next, the inner longitudinal layer is forming the muscle of the bell, which is present in the trigone of the bladder. I hope you have studied about the trigone of the bladder. So in trigone of the bladder, you have not studied about the trigonal muscle of bell. I know you have studied. ठीक है, देखो, repeatedly I am mentioning that you have to study about the urinary bladder. Till now you have not started. ठीक है, एग्जाम में आएगा ऐसे ही बैठे रहोगे. हाँ, तो if I have mentioned that study about the urinary bladder, what is the problem? It is being taught in the class. अगर problem है, notes नहीं है, come and ask. I will share the notes. If required, I will explain क्या problem है. Okay. So this inner longitudinal layer is continuous and it is forming the muscle of bell, okay, which is present in the trigone of the bladder. Next, the waves of the peristalsis pass downwards through the muscle and not depend on the nerve supply. This is how the urine is passing through the ureter. Next, in the ureter, adventitia is present. So as I said, in case of ureter, serosa is not there. Instead of serosa, we are getting adventitia. Okay. So adventitia is outer fibrous coat consisting of loose connective tissue. In that loose connective tissue, nerves, blood vessels, lymphatics, all those things are present. Now this is the schematic diagram of ureter. This you have to draw in a practical notebook. This is the schematic diagram of the ureter. So where is the lumen of the ureter? Star shaped. I hope you can see. So star shaped lumen क्यों appear हुआ है? Because of the longitudinal folds of the mucosa. ठीक है? Now, just now I have mentioned that if you take a tubular structure, उसका lumen का shape क्या है? How you will come to know? TS. LS में पता नहीं चलता है. So I don't know whether you have seen the ureter or not. If you have seen the ureter, thin tube like, ठीक है, you can expect that the lumen will be star shaped. ठीक है, so for that what we have done, we have taken the TS. After that, see, this is the appearance. Okay. So this is the lumen. ये देखो, this is the lumen of the ureter. ठीक है. Now this is the lining epithelium. ये देखो, this is the lining epithelium, which is transitional epithelium. 4 to 6 cell thick. That means approximately 4 to 6 layers of cells are present. Transitional का मतलब क्या है? It is stratified. Stratified means multiple layers of cells are there. मतलब एक layer से ज़्यादा होगा. तो यहाँ पे about 4 to 6 layer of cells are present. ठीक है? After that you can see the lamina propria. ये देखो, lamina propria can be seen clearly. And beyond that, this is the muscle layer. How the muscle layers are arranged? Inner longitudinal, outer also longitudinal, middle is circular. Okay, and beyond that you can see the adventitia. ये पूरा adventitia है. ये देखो, ये पूरा adventitia है. ठीक है? In adventitia we are getting blood vessels, nerves, lymphatics, some area, adipose tissue, etc. So this is how it will appear under microscope, under low magnification. So this is low magnification image of ureter. ठीक है? I hope you can identify the layers. So what I have noticed is identification of slides, especially of urinary system, under microscope is very easy. Okay? सबसे डिफिकल्ट होता है आइडेंटिफिकेशन ऑफ डिफरेंट टाइप्स ऑफ इपिथेलियल टिश्यू, identification of connective tissue, identification of cartilage, then identification of various parts of GIT, okay, especially in case of small intestine. हाँ ठीक है, those are difficult to identify under microscope, that also within 1 minute. Remaining other slide, for example slide of pancreas, slide of liver, then slides of urinary system, those things are very easy to identify. एक बार देखने से समझ में आ जाता है, if you have seen the slide before. Okay, and let me tell you one thing, marks distribution are given identification के लिए half mark and for writing identifying features half mark. Like this the marks are given, means if you are identifying you will be getting some marks. ठीक है, so you have to try that at least you are able to writing 1 or 2 identifying features. ठीक है, for that we will see if 1 minute time is less, we will make it 2 minutes. If 1 minute time is less, we will make it 2 minutes, so that at least you can write something in the exam. Okay, but for that what is important, those points whatever I used to give after each and every diagram, those points are important. Okay, and this type of charts. Next, during the time of theory exam, if the question comes from histo, to save your time, because you have to write and you have to draw the diagram also. तो टाइम सेव करने के लिए क्या करना है, द बेस्ट वे इज, ऐसे जो चार्ट्स होता है, आई होप यू हैव नोटिस इन ईच एंड एवरी टिश्यू आई एम गिविंग दिस काइंड ऑफ चार्ट. तो पहले ये चार्ट लिखो, इसके बाद ड्रॉ द डायग्राम. Okay, next another important point, what you will do in case of drawing the diagram. Now suppose you want to draw the diagram of ureter. ठीक है, now if you draw the entire ureter or everything, it will take time. तो इसका क्या करना है, इसको ऐसे हाफ करो, draw this part only, but everything should be there. ठीक है, so like this what you are doing, one side you are writing the chart, another side you are drawing the diagram, half diagram. ओके, तो उसके लिए थोड़ा प्रैक्टिस करो. विदाउट प्रैक्टिसिंग यू कैन ड्रॉ द हाफ डायग्राम, कुछ नहीं होगा. सो दिस आर सम शॉर्टकट्स बाय विच यू कैन सेव योर टाइम ड्यूरिंग द एग्जाम. Okay, so ureter is done. Next is the urinary bladder. Now in histology of urinary bladder, here also you can see 3 layers are present. Now what do you think, what will be the lining epithelium of the urinary bladder? Same, transitional epithelium. ठीक है. Now if I ask you what will be the lining epithelium of urethra, don't say it's transitional epithelium. पूरा पार्ट में ट्रांजिशनल नहीं रहता है, चेंज होता है. That also in case of male is different, in case of female it is different. ठीक है. तो whenever we'll discuss about the urethra, we'll see there are some changes in case of male and female urethra. Same नहीं होता है. Okay, so don't write that it is same. So internal structure of urinary bladder, supports of urinary bladder, nerve supply of urinary bladder. These are 3 important topics. I want everyone to prepare about these 3 important topics. ठीक है. Next class, maybe in the lecture also I can ask question. Let's see how you have prepared. ठीक है, अगर notes नहीं है तो पूछो, मैं दे देता हूँ notes. Urinary bladder का. I am not talking about histo. I am talking about the upcoming exam. ठीक है. तो everyone, what you have to do, you have to prepare about these 3 topics of urinary bladder. ठीक है. Anytime during the class or after the class or in between the class, I will ask question. I have to check how you have prepared, what you have prepared. Or I may ask anyone to draw the diagram and show me. जैसे मान लो urinary bladder है, draw the diagram of internal structure of urinary bladder, show it to me within 5 minutes. ठीक है, तो prepare accordingly. Now in case of histology of urinary bladder, same 3 layers are there. Okay, if you see the urinary bladder ureter, okay, almost same, some differences are there. Okay, Now the difference, I am not telling that difference is in the lining epithelium. Lining epithelium of urinary bladder, lining epithelium ureter same, but some additional points are added over urinary bladder. Now you have to understand the function. What is the function of urinary bladder? Store, not permanent, temporary storage of urine. Now when the urine is stored in the urinary bladder, when the urine is stored inside the urinary bladder, we know that urine contains toxic materials which are toxic for our body. It is not required, that's why it is thrown out. But for time being, when it is stored inside the bladder, it can cause damage to the tissue of the bladder. It can cause, because इसमें toxic substances है. So in order to protect the bladder tissue from the toxic substance of the ureter, a special type of lipid coating is given to the mucosal surface of the urinary bladder. Those are called as, yes. These are also called as uroplekins. These are called as uroplekins. 2 layer lipid का रहता है, and along with that the cells are attached with junctional complexes. So whenever the urinary bladder is stretched, urine keep on accumulating. So urinary bladder क्या होगा? Gradually it will stretch. So whenever it is stretching, the cells which are there, it will also stretch, and the surface cell will become flattened. जो surface cells होता है, in case of urinary bladder, they are what is the shape? They are umbrella shaped. ठीक है? तो जब stretch होता है, then the thickness of the cell reduces. Because of that, the urinary bladder can stretch easily, and because of the presence of 2 layers of lipid, which are also called as uroplekins, it protects the tissue of the urinary bladder from the cytotoxic effect of urine. Okay, or the harmful or the toxic material which are present in the urine. Okay, so that particular point you have to mention if you are writing about the urinary bladder. Okay, so that is not present in case of ureter, क्योंकि ureter में urine storage नहीं होता है, direct it goes down. Urinary bladder में store रहता है, तो it needs extra protection. I hope it is clear. Now in case of urinary bladder, so 3 layers are there: mucosa, muscle layer, and outer sedosa. In case of urinary bladder, see, the lining epithelium is transitional epithelium, same umbrella shaped. Okay, sometimes it is binucleated. Eosinophilic plaque coat is there, or also called as uroplekin. That can be generally cannot be seen under HNE, but it is present. Lamina propria present, and same mucosal folds are also present. Next, if you see the smooth muscle layer, the muscle coat, it is called as detrusor muscle. Here also see the arrangement, it is same. Inner longitudinal, outer longitudinal, and middle one is oblique. Next, in case of sedosa or adventitia, connective tissue layer is there. Sedosa is only present at the superior part and the posterior surface. Okay, that means the superior and the posterior aspect only covered by sedosa. That means the peritoneum. Rest is not covered by sedosa, it is covered by adventitia. So the mucous membrane is lined by transitional epithelium, okay, or also called as urothelium, which is urine proof, devoid of muscularis mucosae. Surface epithelium is polyhedral. Abundant cytoplasm and prominent nuclei are present. Now if you see some of the nuclei, they are polyploid. The cytoplasm of the cell spread like umbrella. That's why the surface cells are also called as umbrella cells. In case of empty bladder, the mucosa are thrown into folds, and those folds disappear when the urinary bladder distends. Now the lamina propria is there, has superficial dense irregular connective tissue. Now when the bladder distends, the lining epithelium becomes thinner due to the ability of the epithelial cell to change and shift over one another. That's why the thickness of the epithelium changes when the bladder distends. Now this umbrella type of cells having intercellular junctional complex, and it is covered by double layer lipid which are called as uroplekins. Okay? Together they are called as urothelial plaques, and due to the presence of this, it protects the underlying cell from cytotoxic and hypertonic effects of the urine. Okay, so this is very important. So please mention it whenever you are writing the notes of histology of urinary bladder. In case of muscle coat, already I have told you what is the arrangement. Okay, and it is also called as detrusor muscle. What is the function of detrusor muscle? It helps when the contraction is taking place. It helps in emptying of the bladder. Now at the junction, the bladder and urethra, junction between the bladder and urethra, the muscle fibers, smooth muscle fibers are arranged circularly to form the sphincter vesicae. That also you have studied. Okay. And lastly, the serous layer. So already I have told you, superiorly and posteriorly, okay, of the bladder, serosa is present. Rest it is covered by adventitia. Now this is the schematic diagram showing the histology of urinary bladder. Now in case of urinary bladder, if you can see, the muscle coat is very thick. Can you see the muscle coat? पहले यहाँ पे तो देखो, these are the folds, mucosal folds, okay, and then you can see this is the lining epithelium. See, this is the lining epithelium. Next, this is the lamina propria. Okay, below that entirely this is the muscle coat. So this is the inner longitudinal, this is the outer longitudinal, and this is the middle circular layer. Oblique or circular, both you can say. Okay. And lastly, here you can see this is the thin layer of serosa. Okay, lastly, the histology of urethra. So here you have to mention about the male urethra and female urethra separately. Okay, because there are some changes. Entirely it is not same. Okay, so if you see the urethra, it is mucomuscular tube which is carrying urine from the urinary bladder to the exterior. In case of male, it is approximately 20 centimeter. In case of female, 4 centimeter. Okay, you can see the layers. So inner mucous membrane, middle submucosa is there. Okay, which is made up of loose connective tissue. जो submucosa होता है, इसमें क्या रहता है? Loose connective tissue रहता है, and outer muscle layer. So if you see the mucosa, first of all, it is pseudostratified columnar epithelium. Okay, pseudostratified ciliated columnar epithelium, except adjacent to the bladder, which is transitional epithelium. That means जो proximal part of the urethra hai, or you can say the starting of the urethra, that particular part is lined by transitional epithelium. Beyond that, it is lined by pseudostratified ciliated columnar epithelium. Next, at the terminal part, जहाँ पे खत्म होता है, that is lined by stratified squamous non-keratinized epithelium. Okay, so these are some changes in the lining epithelium. Submucosa, as I said, loose areolar tissue. Muscle coat, you can see smooth muscle, inner longitudinal, outer circular, and at the terminal part, okay, what we are getting the skeletal muscle which is forming the external urethral sphincter. Now again you have to notice, so here I have not mentioned about the male and female. Okay, so in case of mucous membrane, especially in case of female, it is lined by stratified squamous non-keratinized epithelium. Along the length, except near the bladder, it is transitional epithelium. इसका मतलब क्या है? In case of female, only 2 types of lining epithelium are there in the urethra. Mostly it is lined by pseudostratified columnar epithelium. Okay. Sorry, no. In case of male, in case of female, it is stratified squamous non-keratinized epithelium, and near the bladder, it is transitional epithelium. That is in case of male. In case of female, in case of male, you can see prostatic urethra, it is transitional epithelium. Membranous urethra, stratified columnar epithelium. Penile or spongy urethra, stratified columnar epithelium. Along with that, we are getting pseudostratified, and next stratified squamous non-keratinized epithelium. So these are the changes taking place in case of male. Female में सिर्फ 2 रहता है. What are these? Mostly it is stratified squamous non-keratinized, and near the bladder, transitional epithelium. That's it. In case of male, so many changes are there. First change you can see prostatic urethra में transitional epithelium. Prostatic urethra के बाद आता है membranous urethra. In membranous urethra you can see it is stratified columnar epithelium. Next आता है spongy और penile urethra, that is stratified columnar After that, pseudo-stratified and then stratified squamous non-keratinized epithelium. Okay, this is in case of male. So these are the changes. So I'll request everyone to note these 2 points in case of female and in case of male. Lining epithelium of the urethra. Next, in the mucosa, certain invaginations are seen. Okay. Why these invaginations are seen? Because of the mucous glands, and these are called as Litter's gland. Now these mucous glands are found along the entire length of the urethra. Okay, some mucus secreting glands are present. These are called as Litter's gland, which are present throughout the length of the urethra. So why these glands are present? Its secretion lubricates the epithelial lining of the urethra. So epithelial lining should be lubricated. That's why it should be moist. That's why these Litter's glands are present throughout the length of the urethra. Okay, submucosa, as I said, consists of loose connective tissue, and muscle coat, same, inner longitudinal, then middle circular, outer longitudinal. Okay, at the lower part, okay, especially you can see external urethral sphincter is present. The membranous part of the male urethra and corresponding part of the female urethra. Okay, जो membranous part होता है of the male urethra, in that particular part, what we are getting external urethral sphincter. That external urethral sphincter is made up of striated muscle. Okay, so what is the meaning? If the section is passing from the membranous part of the urethra, then we will be able to see the striated muscles also. Okay. So that is all about the urethra.